Welcome to this practical demonstration on micrometry. In this experiment, we shall calibrate an ocular micrometer using a stage micrometer and subsequently determine the diameter of a whole mount of frog egg taken as a representative microscopic specimen. The materials required for this experiment are a compound microscope, an ocular micrometer, a stage micrometer, a whole mount of frog egg slide, and a calculator for performing calculations. The ocular micrometer is a small glass disc carrying an engraved scale. It is inserted inside the eyepiece of the microscope. The divisions on the ocular micrometer do not possess any fixed value. Therefore, calibration is essential before taking any measurements. The scale can be observed through the microscope. Since this scale is arbitrary, its value must be determined by calibration using a stage micrometer. Now, the stage micrometer slide is placed on the microscope stage. The microscope is focused carefully so that both the stage micrometer and ocular micrometer scales are visible simultaneously. The eyepiece is rotated until the two scales become parallel. The zero mark of the ocular micrometer is then aligned with the zero mark of the stage micrometer. Further observation shows that 30 divisions of the ocular micrometer coincide with 70 divisions of the stage micrometer. Since one division of the stage micrometer is equal to 0.01 millimeter, or 10 micrometers, 70 stage divisions represent 700 micrometers. 30 ocular divisions are therefore equivalent to 700 micrometers. Thus, the value of one ocular division is calculated by dividing 700 by 30. Therefore, one ocular division is equal to 23.33 micrometers. After calibration, the stage micrometer slide is removed and replaced with the whole mount of frog egg slide. The specimen is brought into clear focus under the microscope. The horizontal diameter of the frog egg extends from 20 to 62 ocular divisions. Therefore, the total number of ocular divisions occupied by the horizontal diameter is 42. For measuring the vertical diameter, the eyepiece and specimen are adjusted appropriately. The vertical diameter extends from 30 to 66 ocular divisions. Therefore, the vertical diameter occupies 36 ocular divisions. The actual horizontal diameter of the frog egg is calculated by multiplying 42 ocular divisions by the calibration factor of 23.33 micrometers. This gives a horizontal diameter of approximately 980 micrometers. Similarly, the actual vertical diameter is obtained by multiplying 36 ocular divisions by 23.33 micrometers. This gives a vertical diameter of approximately 840 micrometers. The average diameter of the frog egg is then calculated by taking the mean of the horizontal and vertical diameters. The average diameter is found to be approximately 910 micrometers, or 0.91 millimeters. Although a whole mount of frog egg has been used in this demonstration, the same procedure can be employed to measure the dimensions of any microscopic specimen, such as cells, pollen grains, spores, protozoans, or other microscopic structures. Thus, using a calibrated ocular micrometer, the average diameter of the whole mount of frog egg was determined to be approximately 910 micrometers, or 0.91 millimeters. This experiment demonstrates the use of micrometry in accurately measuring microscopic specimens. Thank you.